All right, so welcome to our annual PODS workshop. So today our title is really complicated, but really we're talking about DNA methylation, more or less, as you'll see from the next couple of talks. And, okay, this is our schedule for today. So we decided to do this workshop. Our center likes to do a workshop or a symposium every year. And we know a lot of people are working on DNA methylation data, especially with EPIC arrays in either mice or human studies, or other kinds of sequencing data as well. But this is a really popular technology around here, and a lot of people have asked for training on how to use it and stuff like that. So we thought it would be great, even though it's too late for some of you who already learned in the past. But we thought it would be great to have a morning where we talk about the foundational background of why we even study DNA methylation, what the different types of DNA methylation are, what some of the technologies are that are used to measure it, and then at the end dive specifically into these Infinium arrays because they're so widely used by people here and all over the research community, including in environmental health. So we'll have these three major talks here and then a lunch. And at lunch, we can feel free to stay as long as you want. We have two people here from Illumina who makes these technologies. We have all of our leaders of the PODS Core and our leader of the Epigenomics Core as well. And we'll all be here and you can talk to us about any training needs you have, project design, any questions you have, feel free to network with us during lunch. And then for those of you who were signed up for the hands-on, do the analysis workshop, as you know, that's been postponed, but I'll give you the postponed date for that soon. But we already have all the materials ready for it. So it is going to happen probably in the next 2 weeks. So Dr. Yue Zhao is up here with me. We're leaders along with Maureen Sartor of the Omics and Data Sciences Core, which we call PODS. So this is part of our M-LEEaD Center, our NIEHS-funded Environmental Health Sciences Center. If most of you here today are probably either members of that, or maybe your mentor or your lab mates are members of that center. But the goal of the center really is to provide resources for people doing environmental health research so they can do an even better job. And our core specifically focuses on omics, which I'll talk about in a minute. But I think most people here know me, but I wanted Yue to be able to introduce herself too. She's a little bit newer to our center. So,
hi everyone. My name is Yue Zhao. I'm a research assistant professor at the Department of Computational Medicine and Bioinformatics. I have been mainly working on epigenetics data, focusing on epigenetic clocks. And also I have been working on transcriptomics data and long-read sequencing data. So if you have any question or research problem with omics related, you can feel free to reach out to me. I'm happy to help.
Thanks, Yue. And so I just wanted to, just for a minute, tell you guys about what we do. No, this is kind of small, but because for those of you that don't know, we basically provide support, and it's usually or almost always free for helping people with study design, with even doing their data analysis for different kinds of omics data, figuring out the sample size you need and stuff like that. So basically, if you or your mentor are part of or a member of the P30 Center, you can contact us at any time. You can just email Yue and myself or Maureen, and we'll, partner you up with the right person to help you, consultations, work on these questions, give you tips for data visualization, all kinds of things like that. And those are our overall goals as a core, is really to provide support for projects, especially projects with any kind of omics data, genomics, epigenomics, even metabolomics. We have a collaborator who can help with that too. We also try to develop novel data science methods that will meet the needs of new things people in environmental health sciences here at Michigan are doing. And then we also provide training and promote collaborations and networking and learning about new things, which is by doing workshops like this. And these are just some of the things our bioinformatics teams have worked with, of course, genomics, but I'd say we actually do quite a lot with RNA-seq, with the EPIC arrays, but with, whole genome bisulfite sequencing. Maureen has developed technologies to analyze ChIP-seq. We've got experience with all of these, and the stuff on the right are kind of the new things our PODS Core is getting into in the last couple of years. So now multi-omics is a big deal. So, our core is getting more into how you incorporate different omics data sets and maybe we'll have a workshop on that in one of these future years. So with that, we would like to bring up Claudia Lansett, our wonderful director of the U-M Epigenomics Core, who's going to tell us all about DNA methylation and what this core can do for you. Thanks.
I've not even talked yet. Wait a minute. Okay, so I'm just adjusting. So, good morning, everyone. I will not reintroduce myself. The weird accent is I'm French Canadian. Okay, so let's clear that out. And it's a pleasure for me to come and meet you today and discuss DNA modifications. It's going to be, for a lot of you who do DNA methylation or some epigenomics, it may be a refresher. However, it's always good, just like some of those trainings that the university makes me, but probably some of you do all over again every year, and we're always like, oh yeah, I didn't remember that. But it's also important to have good foundation so that it helps, not when it goes well in the data analysis, but when there's something going wrong and you need to troubleshoot. It's good to have some of this background fresh in your memory, or being able to know where to go and check things and remember. So we'll set the stage so that we're all on the same page as what epigenomics is, because it can mean different things to different people. And what we talk about when we say epigenomics layers, so which will be the marks. We will focus on DNA modifications for this, because this is what we analyze with the EPIC array, and this workshop is meant to be for EPIC array analysis. Then I'll go a bit into how we do the measurement. Some of you work with the data, know that it involves a certain treatment, but I've never really done it. So I will go into the basics as to why we treat the DNA with certain reagents or use certain kits and enzymes. There's new methods. So I will briefly talk about that. I'm not going to spend too much time on the epigenomics Core, but I've been asked to, just say that we exist. We've been here for 10 years. So I'll just briefly tell you all the other things that we can do. And as Jackie said, I'm here all day. So if you have any questions, don't hesitate. And for the people online, there's going to be an email address. So if you do have any questions, you can reach out to us that way. So let's just go with what we do when we study epigenomics. So we study changes in gene expression most of the time when we're looking at changes in cell expression or cell functions. These changes will be heritable and stable through time, and they will not be caused by a change in the DNA sequence. So these, especially for DNA modifications, they're required for life. So if there is any factor that impacts DNA modification, especially DNA methylation during early embryo development, this is lethal. It is necessary for forming a new life. Heritable can be through cell division. So changes in these epigenomic modifications can be communicated at cell division, so from mother to daughter cells. It can also be heritable through generations. Dutch famine, I think, rings a bell for everyone. And there's also some other historic events that have been studied in that sense. And we're also now starting to help investigators that are interested in different other changes, environmental changes that I will touch on later. So basically, it's just a mechanism for the organism to adapt to environmental changes or signals coming from the environment so that they can survive, basically. And that's important because, as you all know, every cell in your body has the same genomic code. Every cell has the same sequence of DNA. So, but each and every cell uses a different set of genes that are expressed to become a muscle cell, a heart cell, a brain cell. And that is being possible through epigenetic information. This information is plastic, it's dynamic. Some of it is set and may not change at early embryo development. There's also, as you all know, two major waves of demethylation that occur early in embryo development. But some of these epigenetic changes will also change throughout life. And we know that there are changes in some DNA modifications that occur as we age. And that is actually the foundation for the interest in all of these epigenetic clocks that we will discuss later on. So these changes are essential not only for the organogenesis, like development of our different tissues, as the fetus develops, gamete biology, you all know about imprinting. It's also essential for memory and learning. So those are natural, healthy, if you want, ways epigenetic is helpful for adaptation and reprogramming. However, sometimes there are some signals that will come and the adaptation will not be preferable, but essential. And these will also be associated a lot with cancers, also been associated with heart disease, imprinting disease, of course, but autism, depression, there's been linked to asthma and a whole set of other disease. And some of those can be said during early life, embryo development, or throughout the life, some natural changes. But throughout our lives, we will also get different cues from the environment as like you're learning now, or you're recreating and, stratifying, solidifying some pathways that that you already knew, but the knowledge is coming. But stress will have an impact, lifestyle, and also nutrition. So pizza may not be good association with your epigenome. I can't say. But also the air, the environment, pollution. I have a group from Kalamazoo that is looking at the impact of climate change on early embryo DNA methylation. They've also looked at the impact of stress from some Bru tribes where protecting the food, so the impact of being protecting food versus versus men who are not doing that have a consequence on their epigenome. And there's also a lot of work that's being done at ISR on social economic factors and also early childhood adversity. And that's been a lot of work that's been done. Environmental toxicants, which here is the plasticizers. And also some medications will have an impact. So all of these are coming together to send signals to the system, the organism, to adapt. Now, when we talk about epigenetic, like marks that transmit this information, of course, there's DNA modifications, and we're not going to talk about this today, but histone modifications are also of importance. These are the two major marks. And so DNA modifications, we're taught mostly from 5-methylcytosine, and we'll touch a bit on hydroxymethylcytosine later. These marks will be more stable. These are the closest to the DNA, if you want, because they are directly being, they're a direct modification on the cytosine residue on the DNA. However, they're not changing the cytosine into anything. It's still read as a cytosine. They will affect the DNA structure. So they will help compact or relax the conformation of DNA. And they've been associated with development and printing, tissue expression, and some marks like hydroxymethylcytosine are associated with increasing chromatin accessibility, being poised, like chromatin being poised to be expressed, transcription factors coming to and other chromatin modifiers. Whereas histone modifications are more transient when you think about it. So the histones, as you know, are part of the nucleosomes. That's how the DNA is all structured. That DNA is looped around those 4 histones. And they have tails that kind of come outside. And these can be post-translationally modified by enzymes, either by methylation, acetylation, ubiquitination, phosphorylation, or just a few. There's many more. So these can be changed. You can have phosphorylation, dephosphorylation, acetylation, deacetylation, etc. And these are more rapid and can be modified and get gene activation or repression. And they've been associated with differentiation, DNA repair, DNA damage. And how these work, and they work together, is basically just to control how chromatin is accessible. So how certain genes in certain tissues will be amenable to transcription with the right factors around. And how, so histones will further, when you have repression, just really compact. And we got this huge DNA that has to be all compacted into this tiny nucleus, and it needs to be available at the right time in the right place. So they work together by recruiting chromatin modifiers that can either further condense the chromatin and make it ethyl chromatin, or recruit other modifiers and transcription factors of transcription machinery that can activate genes. And also, there was a new study recently where actually hydroxymethylcytosine deposition, or not deposition, modification comes before chromatin opens. So it can be a predictor as how chromatin will be changing and become more accessible in certain cell modifications. So, so in terms of DNA modifications, we're going to talk briefly about animals for the rest, because the EPIC arrays mostly work with human and mouse. However, it's not just animals that do have epigenetic marks and that use epigenetics. Actually, DNA modification have been discovered in bacteria. I'm a nerd. I always like to put these little tidbits there. I'm always asking lots of questions about like, where does this come from and everything. So bacteria, but also archaea. And they use it for different not reasons, but purposes. But we're just going to focus on 5 cytosine methylation. So you've seen this before. And this is to show you basically that, yes, we all look at DNA modification, like DNA methylation. However, it's not the only modification when we talk about DNA modifications. Now, you will not find all of these in lots, in high proportion. One thing I want you to always keep in mind is that the only mark that you can actually deposit on a cytosine is the methylation. It's the methyl group. And that is done by DNA methyltransferase, either de novo by DNMT3s, or when the cell is replicating, and then there's the maintenance DNMT1 that comes and deposits the methyl group on the sister strand at division. All of the other marks for 5-hydroxymethylcytosine, formylcytosine, carboxylcytosine are all modification through oxidation of the DNA methylation. Now, another thing you need to keep in mind is that, you know how like anyone who's done biochemistry and enzymology, like a lot of times like phosphorylation, you will have one enzyme doing one thing, and then you'll have another enzyme coming the other way around. So phosphorylation, then there's another enzyme that can do dephosphorylation. It's not the case with cytosine modifications. You cannot go between a cytosine and methylcytosine and come right back with one enzyme. If you look here, it's through successive series of oxidation, and it involves the DNA repair mechanisms. This has consequences because errors can happen there. So that's why sometimes there's going to be some SNPs that are created by some of these at DNA repair when there is change from cytosine to from fibrinosyl cytosine through cytosine back. Of course, there's another way. So if you cannot do the maintenance at division, if you do not have the machinery to copy the methylation, and that happens in early embryo development. There's in the maternal where there's like a passive demethylation where the methylation is not copied to the other strand. However, when we look at active demethylation and we go from methylation to hydroxymethylation, then the only way you can come back to the cytosine is through oxidation with DNA repair. Now, 5-methylcytosine is the modification that you will mostly find in tissues and cells. The other one is going to be hydroxymethylcytosine, but as you can see, the proportion is not very high when you compare it to cytosine modifications. It's also not really found in every tissues. Neurons, placenta, early embryo development, cancers, depending, will be the cells, tissues where you will have most hydroxymethylcytosine. So when you're looking at which marks to look at, depending on the tissue or your question, of course, if you're doing something that's impacting the oxidation, like the TET enzymes, then you may want to look at changes in hydroxymethylation. However, depending on the tissue, methylcytosine may be the mark that is of best interest for you. Where do we find those? Well, we find them, as you know, not on just any cytosines. Plants will have any cytosines that is becoming methylated. In humans, that's not the case. We find them in a specific context, which will be a cytosine followed by a guanine, so what we call a CpG dinucleotide. And these are not randomly distributed across the genome, and they tend to cluster in what we call CpG islands. They can be found in regulatory elements mostly, or inside genes. So we'll have this regulatory elements like enhancers. We can find them. You'll have a lot of CpG islands are found in regions that also correspond to promoter regions, like early or different enhancers or promoters. You will find some of them that are not necessarily in clusters throughout some gene bodies. Sometimes it's an intronic promoter or an intronic enhancer, not always. And we find a lot of them in the repeat elements. That's probably where most of the methylation will be found, because we don't want these sequences to jump around and create havoc within like the cell context in cell functions. Some of them are also found in what we find orphans in intergenic. That's kind of a humanized representation of the genome that we have. So they're not in genes, they're between genes. So that's why we call them this way. In some cases, we don't know why they are found there. Maybe because we don't have enough knowledge of, what if there's a function to these. There's increasing information of DNA methylation being also involved in the chromatin organization in where CTCF can sit. CTCF does not like methylation. So depending on where the regions are, that can also be why we can find some of those there so that you can have all of those topology regions. And so usually when you have methylation, especially in regulatory elements like promoters and enhancers, it's been associated with silencing, which kind of looks weird to us when we think like, well, what about if we have DNA methylation within gene bodies? Then it can bring some other machineries and help with splicing, or actually, I'm going to go to the other one, with splicing, or just recruit different other chromatin modifiers that may actually come and impact histone modifications to help get expression. As much for hydroxymethyl cytosines, they tend to be found in enhancers in gene bodies for the actively transcribed genes. And again, where chromatin will be becoming like signal for the machinery to come and just like modify this chromatin here to make it permissible for transcription. Now, how we study it, there's a couple of ways we can do it. But today we're going to focus on the EPIC array. And I'm not going any further than this in terms of how it works with the EPIC. We've got the experts in the room. I'm going to focus more on what we call cytosine deamination and amplification. It's going to be very brief. But basically, we take the DNA, we treat it with enzymes or chemicals, mostly chemicals, to it to be able to distinguish methylation modification to non-modified. And then we do amplification on it, and I will show you why. Then that DNA gets fragmented and then resuspended into a hybridization buffer that then we put this with the EPIC array. Once it's been hybridized for a certain amount of time, there's washes, then there's some extension, and they're going to go on this and staining, and then we scan. Well, I say we, but it's more like my colleague at Advanced Genomics Core that does that for me. I do the first step for you. And that's how you end up with the spreadsheets that we all know and love with all the data and the measurements and the signals for the different CPG sites, okay? So you've all heard about probably bisulfite conversion. If you've done EPIC arrays, either you've done it or I've done it for you. And that's considered the gold standard. That's how we've been able to really get study methylation, basically, for like, and differentiate it from non-methylation. So basically, it's a chemical reaction, sodium bisulfate, where the cytosine is being, make it simple, goes through some chemical. I'm not an organic chemist, I'm a biochemist. So just goes through some stages of chemical reactions to be converted into a uracil. And the methylation and hydroxymethylation, oxidation of that methyl group, protect the cytosine. So that's why only the cytosines that are not modified can be converted. Why we do amplification or PCR reactions? Because that's the way we can identify which cytosines were modified or not. Because when we take the DNA, it's been bisulfite converted. So here you see those cytosines are methylated, and some other ones here, they're not. So they've been converted to uracil. So when the polymerase goes and reads this, it puts a complementary, you know how it works, right? Puts a complementary nucleotide on the other strand. So when the polymerase sees a uracil, it will put a thymidine, like a T, on it. And basically what we're looking at when we are by sequencing or by the arrays, what we are looking at is a C to T change. So it's a C to T SNPs. So that's how we distinguish cytosines that were modified from, so cytosines that were modified will remain as Cs or genes on the other one. And then these ones will go as Ts. So there's advantages to this because this is what gives us base pair resolution to measure methylation at specific sites. And it gives us a pretty precise quantification. I would think that the only other way you can have that precise quantification is by mass spec. But here, not only it gives you a quantification, it tells you where it is in the genome and the level. However, it's got limitations. If there's anything that goes on with the bisulfite conversions, if it's incomplete, or if the desulfonation, like in the cleanup stages, something is not complete, it will not give you a proper conversion. So you will have some false positives with it. As I've shown you, because methyl cytosine and hydroxymethyl cytosines are both protected from it, you cannot distinguish them. So in some tissues, it may not be that important, but if you're looking in brain, neurons, or you're doing a treatment that you know is going to impact hydroxymethylate, like methyl cytosine modification, that can become a problem. And if you're looking at amplification for targets, looking at specific regions where there's a potential change in methylation, well, you've got to be very careful when you design your primers, because you're basically going from a 4-letter ATGC genome to like a 3-letter, because you've lost most of your seeds, which were not methylated. So there's ways to address these two specific limitations, and I'm just going to touch base on them. And now the next slide, Christine may grimace, okay? She knows what's coming because she's done it. So one way you can distinguish methyl cytosine from hydroxymethyl cytosine is by adding another chemical before you do bisulfite conversion, which is pertinent here, this really complicated one. And no, I don't know it by heart. I actually wrote it on my notes because I can never pronounce it. It's actually made a mistake. So you take your DNA and you chemically oxidize the hydroxymethyl cytosine to make it become like a 4-mil or like a 4-mil cytosine. And then you do bisulfite conversion, which is actually like it's deprotected. Your hydroxymethyl cytosine is not protected anymore and is going to be converted to a uracil. And then when you do the amplification, like the PCR steps or linear amplification, depending on if you're going on the array or if you've been doing like sequencing to go on the sequencers, then your hydroxymethyl cytosine here is going to look like it's seen. Now it has advantages, especially if you're interested in methylation and you don't want to know about, you want to be sure that what you're looking at is methylation. Well, oxidation with bisulfite sequencing is going to give you the real measurement of methylation. If you're interested in hydroxymethylation and methylation, well, then you need to do both bisulfite conversion and oxidation followed by bisulfite conversion to get the two measures. And no, you cannot do subtraction. That's how you get negative values. There's no such thing as minus hydroxymethyl cytosine, but there's an R package. That's what we use in the core called the MLML2R. that will take the results from both assays and give you an estimate. It's maximum likelihood methylation levels. That's what the MLML stands for. And based on the data, the measurements from both assays will decipher how much methylation, hydroxymethylation, and no modifications. It will give you all three. And one thing though is that bisulfite on its own can damage your DNA. Adding oxidation makes it worse. So it adds a further damage to it. And you need a bit more DNA sometimes to leverage for that. And because you have to do two assays, then it's like you need to have twice the amount of DNA going into each reaction. And then, you put both on their specific place on the EPIC arrays, but then you get the data. We've actually done this for Jackie and some other colleagues, where they have successfully used OXBS to look at changes in modification for PFAS exposure. Another way that is less damaging for the DNA, and that's a new kit that have come like a couple of years ago, is using an enzyme called ApoBec that is known to do deamination enzymatically. So that's not as damaging on the DNA. And there's a way to tweak it. So basically, when you do what's called the EMC, it's from MEB, if you do what's EMC, then you're going to have oxidation with a TET. And the enhancer is basically the glucosylation. There's no big reveal here. And when you do these, it's going to go protect both your hydroxymethylcytosine, and after oxidation of the methylation, these two will get beta glucosylated, and that protects from ApoBec to deaminated. That you can see here. And then after deaminate, like when you expose the DNA to the enzyme, then any cytosine that is not protected, so it's going to be the cytosines that have no modification. It's an enzyme. It's got kinetics, so it's going to take, its preferred substrate is going to be cytosine that is not modified. If you leave it long enough, it's going to demethylate your methyl cytosine. It's not going to do anything to your hydroxymethylcytosine. In fact, you would not need to do beta glucosylation on it, but sometimes, things happen. So for good measure, we do beta glucosylation on the DNA. And that's actually one way, if you look here, how you can go and get a specific, direct measurement of hydroxymethylcytosine in samples. So this way, you do not have the oxidation. You go straight into glucosylation, which is going to protect your hydroxymethylcytosine. You don't need to leave it longer. And we'll leave it overnight, for example. And then the methylation will get the cytosine methylate, the cytosine will get deaminated, and then by PCR. So this is one way that you can have a direct measure of hydroxymethylcytosine. So let's say you want to know about changes in hydroxymethylation within neuron development or something. And either you already have methylation data, or you just don't care about it. That can happen. Then that could be a way, just a 5-HMC. And these, sorry, I'm jumping. So that's one way you can have it. Again, you need to have more DNA because you need to do two assays again. So an MLML tool can also do that, decipher, if you want to have methylation and hydroxymethylation. Because remember, when you just do the regular, like bisulfite conversion, the enzymatic deamination by itself is still giving you a readout of both methylation and hydroxymethylation. Now, until recently, and I have tried to do this, I couldn't get this to work on the EPIC array, but someone figured it out, and they actually even showed that you can use this with cell-free DNA. So you can put cell-free DNA on the arrays, and they've actually went and did some single cell, like really low input of cells into this. So basically, they do the enzymatic kit, the EM-seq, and then instead of putting this on the sequencer, they put it on the array, basically, right? So I haven't tried this. Thought I was gonna try it for cfDNA, and then like they changed their mind. So if anybody is interested, have questions, come see me. I really want to try. So that's pretty much what I have for you as background or refresher, depending on where you come from. Now, may seem a bit complicated for some of you. My staff refuses to take pictures. I am the only one who's stuck and forced to take pictures from my core. And I hate it. I'm the one grimacing now. And it was during the pandemic. So, well, after. So you can come and see us. If you have any questions about what we have time today, but if something pops out in your head later on or anything, you can come see us because that's what we do. That's why I came here 10 years ago to help with. We can do consultations. We will help you for the array, histone modifications, anything. And we will work with you. We do now work with the bioinformatics core for anything analysis, but we'll have consultations, help you. I mean, you're lucky because you've got the pods. And so when we know where the data is supposed to go, we will transfer it straight to the appropriate person for you. No problem. We always do QC on what we work on. So then they will get the QC on all the files. More than happy. So as you know, epigenomics is complicated. I'm going to go through all the list. I just put it there because we do a lot of cytosine modification, but we can help you with all of the other ones. And we don't own sequencers. I like to repeat this just because some people think that they come to us and then we'll sequence it. And no. We have one of the best sequencing cores, the Advanced Genomics cores. I've worked with many sequencing cores. I'm old. I've been doing this since the G2AX. And we have one of the top-notch sequencing cores, and they don't pay me to say this. Like, I work with them, and I've worked with different ones. So we're lucky enough. And so we partner with them. They give us the data back, we do QC, and then we give it back to you, or bioinformatics, or M-LEEaD, depending on who's going to do your analysis. And we're at MSRB too. So thank you. And if you have any questions, I'm happy to take them. Or if you want to wait until later, no problem. Go for it.
Have you had many investigators do single cell epigenomics yet? And which taxi methylation?
I don't handle that bit. Advanced genomics and develop with 10x. However, I can help with nuclei isolation because we do it for for ATAC. We have to extract nuclei if it's bulk. We are onboarding the single cell methylation. I've got actually my first project. I'm just waiting to have time to be in the lab. It's the Scale Bioscience kit, which we've tried. So we're onboarding that right now. That's going to go through sequencing. But yeah, I'm happy to chat. There's a spatial epigenomic platform. It's not really a machine because it's like thingies. It's a bit more hands-on than like 10x or other things from Atlas Omics. That is also waiting for me in the lab. So basically you can do ATAC and you can also do cut and tag on a piece of tissue. So we've been trained for it. I'm just trying to finish implementing it. So that may be the next thing I come to present to you. But yeah, that, and if we just take if we isolate nuclei for you, then we'll bring them over to HEC for you. We're not going to give you nuclei for you to go. Like we will schedule the appointment to bring it over. So I call my cousins. It's like big family.
Okay, so this is one application that I know a lot of people at University of Michigan are also using the clocks for. Epigenetic age is very popular now, and this is something that can be calculated from the data, or really primarily just from the data on these arrays. And there's even versions of it for the mice too, but it's mostly using humans so far. So we're lucky today we have Tina Lee here, a graduate student in bioinformatics, who also is one of our PODS kind of staff scientists as well. So if you ever get an analysis done at PODS, Tina might be one of the people working on it, and she's put together a really great explanation of the clock. So thank you, Tina.
Oh, thank you. So, hello everyone. Happy Friday. I'm Tina Lee from Marines Lab, and I'm a 30-year PhD student. So from the last paper we published, we have do many epigenetics clock analysis, and I think it's really fun to working on this for our data set. So I'm going to share about this interesting tool today. So today I'm going to go through the definition between the biological age, chronological age, and what is the epigenetics clock, and the 5 most common epigenetics clock we're using in the research. And I'm going to talk about the age acceleration, and then I'm going to go through this really interesting small paper which they investigate the household air pollutions with the epigenetics aging. So first, biological age is different than the chronological age. So the chronological age is the number of years you live. So for example, this year I'm celebrating my 24th birthday, but I cannot celebrate the 18th birthday next year. So this cannot change. It's the number of the candle you put on the cake. But biological age is more complex. It's the measure of how well the functionality of the body. So for example, when you are 21st years old, you will feel okay, you can digest the alcohol and the sugar pretty well. But when you get 60, you may have a little bit problem to digest alcohol and the sugar. So this is from the biological aging. I would like you guys to come up some of lifestyle behavior that can help to slower the biological aging. So anyone have any? Yeah, exercise. Diet, yes. I just read a really interesting paper about eating the oatmeal diet can help to decrease biological aging. And
Meditation.
medication. You think that will increase or
Meditation, not medication.
meditation?
Yeah.
Okay, sounds interesting. Yeah, actually there's a lot of things can impact biological aging, like genetics and epigenetics factors, but more from external, lifestyle, how much you exercise, and also the diet, how much red meat you eat, how many vegetables, fruit you are eating, and also it's from environmental, so the UV light. Always there's some dermatologists wear sunscreens can help to prevent aging for your skins, and also air pollutions. Also the disease and the treatment. There's many age-related disease. So maybe you can think of people commonly most of people catch the cancer around 60 than at 18. So that's also something about biological age. I would like to also point this person, Brian Johnson, because he's a millionaire, spend a lot of money every year to have a team for him to do anti-aging, and he claimed himself is 18 in the biological age. I think that's really crazy. And also I just read a news recently saying he is launched a contest for the people who are really confident. If you are really confident about I'm younger than like the people who are in my age, you can just like provide your blood sample and you can join the contest. And if you like to see like whether you have like the lower like age accelerations and biological age accelerations. So thank you for Claudia to have like introduced like the DNA methylations. And as I mentioned in the last slide, the biological age is actually really complicated to measure. However, there's like some bioinformatics approach that's developed that can help us to measure the biological age from the DNA methylation fingerprints. So actually there's like a theory behind the epigenetic clock is by the patterns in the aging, which is the DNA methylation is lost in most genomic regions as people age. But in specific loci, like the CPG island or the promoter, the DNA methylation level is increasing. So we can use this pattern to build a model and to train a model and have the epigenetic clock that can like predict the chronological age. And the DNA methylation alternations are also linked to the age-related disease and the longevities. And this highlights their potentials as a therapeutic targets. So for example, like our lab, we are doing something about the amyotrophic lateral sclerosis disease, which is ALS, and famous for, and this like, and the Stephen Hawking and the Lou Gehrig are getting like this, this disease. And so like we are, our paper just highlights that like this disease can related with the biological age accelerations. And also there's like many other age-related disease like the Alzheimer, like the cancer. So epigenetic clock is just like really get like really popular to study in this like age-related disease. So this is like a 5 common human epigenetics clock that are using in the research, and they all come from the same labs, from the Harvard labs. And actually there's like 2 generations of epigenetics clock. So the first is the Horvath clock and the Hanna clocks, and the second generation is the phenol age skin and blood clock, and also the green mate, I think. Yeah. They cut off my tables. Yeah. So for the first generations, which is the Harvard clock, this is the first published like age estimators, and they shows the evidence that the epigenetic clock can strongly related with the chronological age. By their correlation is like 0.96, and they are like the multi-tissue age estimators, which they are training on the TCGA TCGA Atlas healthy tissues. And they can adapt in Illumina 27K and the 450K arrays. So this is like a three correlation plots that show in the paper, which in all tests, the performance is pretty well, it's 0.96. But in the blood CD4 T cell samples, the correlation is only 0.78. And also the performance is bad in some of the tissues, like the breast tissues. So that's all like why they just like updating, like they keep updating their clocks in their lab. The next is the Hanna clocks. So unlike the Horvath clock is on the multi-tissues, the Hanna clock is focusing on the whole blood tissues, whole blood samples, which they are using the 450K arrays. The correlation is 0.96, and the median absolute difference is like a 3.9. However, there's still like a weakness for this clock. It has like a bias to estimate the age in the children populations. But the Hanna clock have like a higher prediction accuracy than the Horvath clock in the blood, which is 0.96. Like the previous one, the blood CD4 T cell sample is 0.78. And in this paper, they first show epigenetic clock can use to study the cancer. So on the left panel, the red dot is the tumor tissues, and the black dot is the normal tissues. You can see the tumor tissues will have like slightly higher than the normal tissues, which means the tumor tissues will have a higher age acceleration than the normal tissues, which I think is like really interesting to show this evidence at that time in the Hanna clock. The next is the phenol age, which this is the first age estimators that shows the clock can have like can shows like a weak correlation with the lifespan and the clinical features. And this is can widely adopt in 3 chip platforms, 27K, 450K, and the first version of the EPIC arrays. And it shows that it can predict the 10-year and the 20 years mortalities. So if you look at the left panel, which is the hazard ratio models, which indicate for this clock as a 1 year increase in this in the DNA methylation phenol age is actually associated with 4.5% of increase in the risk of death. So they also this shows like stronger association with the lifespan than the Horvath clock and the Hanna clock. The next is the skin and the blood clock. So this one goes to the second generations. So they start like the clock start to can predict the like lifespans and the clinical features. And this is like updated version of the Horvath clock because they're in the like they can adapt it in like multi-human samples like the skin, blood, brain, neurons, glia, and the livers. And also this is the first like clock that can detect the epigenetics age accelerations in the segmental progeroid syndromes, which the patients will exhibit a dramatic accelerated like development. So I would like to show the like updated like performance for this clock. So in the fibroblast samples, the Horvath clock prediction is like a little bit bad. The correlation is only 0.71. However, the skin blood clock, the correlation is 0.91. And the most important is in, and it's a stop. Okay, so most important is in the prediction in the blood samples. So in the for the skin and the blood clock, the correlation is 0.98. But like in the Horvath clock and the Hanna clock is 0.96 and 0.97. So they update they improve the performance of predict the age. The next is the Grimm age, which I think this is like really important clock and is like widely used in the research. By this clock can strongly predicts the lifespans. So that's like the reason why like many like disease study are using this clock. So they're actually construct their clock by 2 steps. So first, they're building the elastic net model for the 88 plasma protein variables with the DNA methylation, with all the DNA methylations beta values. So from the elastic net models, they only find like 12 out of 88 plasma proteins exhibit the strong correlation with their DNA methylation based surrogate markers. So we're going to use this 12 plasma proteins in the next step. And for the second step, they're building the elastic net Cox model by using the time to death and regress out the chronological age, sex, and the DNA methylation based biomarkers of smoking pack years and the 12 selected plasma protein levels. So this result remains like 8 plasma protein levels, age and gender and smoking pack years in the model. So they're using this for like for the clock to build like the linear model. So the Grimm age basically is like all of this, the clock is in the linear model, which is like the weights times the CPG's beta values, and then we sum them together to get the Grimm age. So the Grimm age can strongly predict the lifespans. So for the time to death, all-cause mortalities, it shows like as a 1 year increase in the DNA methylation Grimm age is associated with 10% increase in the risk of death. And for the time to coronary heart disease has a ratios, it shows as a 1 year increase in the DNA methylation GRIMAGE is associated with 8% increase in the risk of getting the heart disease. So this is like examples of like how GRIMAGE can study like the lifestyles, for the clinical like research. And not only like the GRIMAGE can be used in studying like the clinical stuff, but also it can like they are associated with the diet, education, and the lifestyle factors. So it's really interesting to see that for here, you can see like the fruit and the vegetable have like negative correlation with the age accelerations. But like the fats are having like the positive correlation with the age accelerations. And for the lifestyle, we can see like for the smoker, like you have really strong positive correlations with the age accelerations. So yeah, this can also adapt in like many like sociology studies. And I would like to give like some of example scenario to use the good epigenetics like a clock. So for the first conditions that we have an Illumina 27K DNA methylation data set from the human blood, and I want to impute the chronological age to be included in the analysis. What do you think will be the good clock to be used for this case? So that will be Hannah's clock. And for the second one, I have like a 450K EPIC arrays for the human whole blood data set for both the patients and the healthy controls. And I want to conduct like survival analysis between the patients and the healthy controls. What clocks you think it's suitable for this case? Yes, GRIMAGE. And if I have like Illumina EPIC DNA methylation datasets from both breast tumor and the healthy epithelial tissues, and I want to study the age acceleration between these two tissues, like what's the clocks you think we should use? Yes, it's really easy, right? Yes. So basically, like if you have a study and you want to determine like what's the perfect clocks to use, you have to answer like 3 questions. First is like what's the array type of your data set? Second, is like what's like your aims for study, like survival or chronological age. Third, which is what's like the tissues like your data set are from, and like what the species of your samples. So these 3 questions can help you to navigate to the right clocks to use. And epigenetic age accelerations is actually the biomarkers for like many like disease and like lifestyle like studies. And this is actually measure like how much a person's like biological age is older or younger than their chronological age. However, we cannot do in the age acceleration by the subtraction from the chronological age by subtract by the biological age, because, for example, the GRIMAGE correlation is 0.82. So this will maybe like unfair for the clock to which strongly predict the lifespan. So we are going to use the residual epigenetics age accelerations, which the REAA is going to be calculated by the residuals of the linear model of the chronological age versus biological age from the healthy controls. So, for example, the plot I'm showing. First, I'm going to calculate the gray lines, which indicates the linear model of chronological age versus the biological age from the healthy controls. And for each dot, which is each samples, I'm going to calculate the distance of like this point to the gray lines. Next is I'm going to share like a small paper about investigating like the household air pollution effect with the epigenetics aging. So the background about this paper, the household air pollutions from the indoor combustion of solid fuel is like a global health burden linked with the lung cancers. Polycyclic aromatic hydrocarbons are produced from a combustion of the smoky coal and are significantly like associated with the high risk of the lung cancers. The 5-MC is one of the example of the PAH, and as like studied before and approved as proved to be associated with the risk of the lung cancers. So that's like why we need like DNA methylations to study from between the air pollutions and the risk of the lung cancers and also the aging. Because the DNA methylation may be influenced by the environmental factors, and epigenetic age may reflect the environmental induced effect on aging, which is also another major risk factors for the cancer. So for the method, they collect 106 never smoking women, and the field types for each participant is collected by the questionnaires. And they use the validated exposure models to predict the level of 43 household air pollution exposures. So they're going to study the association between the exposure clusters, household air pollution exposures, 5-MC, with the risk of the lung cancer and the EAA going to be tested. So first, they test 5 clocks associations with their data types. So also this is something I recommended for start. Like you can test all the clocks and to see which one have stronger correlations and fit with your data set as the first steps. So you can see that for the gray image, other than the other 4 clocks shows significant correlations with the fuel types. So and they by the way, they set the smokeless coal as the reference. So you can see they all have positive correlations with the smoky coal wood. And you can see the difference between the current fuel type and the childhood. The childhood may have higher correlation than the current fuel type. And also they test the association between the HAP clusters in the current childhood and the cumulative status with the EAAs. So they also test the IEAA and the EEAA, which the IEAA is the intrinsic EAA, which are independent of the lifestyle factors and the immune cells. The EEAA will count both the intrinsic cellular aging and also the influence lifestyle factors and the immune cell compositions. So from this plot they're showing, I think it's really interesting. Still a green image shows strong correlations with the exposure clusters. And you can see there's 7 of clusters are associated with a higher EAA, but there's even 2 2 HAP clusters are associated with the lower EAA. There will be I think it's the sulfide dioxide and the DIP. So this is really interesting. And also they test the associations in current childhood and the cumulative status for 5-MCs and the EAAs. So still green image are the one that shows strong correlations, and it looks like the childhood exposures for the 5-MCs will have higher correlations with the with the green image. So something you can take home from this paper. So this is the first epidemiologic studies that suggest that exposure to the indoor air pollutants is associated with the accelerated epigenetics aging. Cumulative 5-MC before the age of 18 was associated with a higher lung cancer risk compared to the adulthood exposures. Exposure to the PAHs in particular during the childhood, may be the contributor to accelerating aging. Higher grammage EAA for never smoking women exposed to the PAH from using smoky coal for cooking and heating in the home. And in the paper, they didn't show the survival analysis, but like age acceleration, the biological age accelerations is also like a powerful to doing like the survival analysis. So this is from the paper that we published recently. We shows that the people who have like a fast EAA accelerations may have like worse post diagnostic survivals. But like the fast aging group, which is the red lines, have like worse survival times. So the median of survival time for the fast EAA group is like 1.81 years, but for the normal and slow EAA is like 2.44. And on the right panels, we associates the age accelerations with the exposures in the ALS patients, and we find there's like 5 exposures are correlates with the age accelerations. So not only there is like a human epigenetics clock, there is also like non-human epigenetics clock. I think like a Harvard have like a mammal clock array or something that are available in the Illumina that can test for like the mouse and many like species. Yeah, that's all for the epigenetics clock. Is there like any questions?
Which clock did you use for the ALS analysis there, observation?
So we are using we are mainly test on like the brain age, but we also test for the HANA and the phenol age, and also we use the other one that are not from a Harvard plot, which is the Doodling poem. So they both shows like really like strong correlations, like strong evidence in the ALS disease. But we are using like the grammage because this is like widely used for disease study.
So for your studies, are you developing a clock of some sort or just using those clocks with your data sets?
That's a great question. So we are using the grammage, but like our next step, we're thinking of like develop like maybe like epigenetics clock for specific for the neurodegeneration disease. Because like there's like many like I think there's like some like clocks for specific like study like liver, and then we think okay maybe like from this from like our finding from the last paper we can like build like epigenetics clock specifically for the disease. Okay, yeah, thank you.
Well, thank you too. Thank you to all of our speakers today for coming out. Thanks to all of you for being here as well, and to the people online. Our formal session is now done, but I'd like to thank Epigenomics Core, M-LEEaD, and especially Rose, who's setting up our lunch, so she's not over there, but for really making this happen and getting us good food and coffee. So we have